The pyruvate dehydrogenase complex. This is a group of enzyme and coenzyme which gonna work on pyruvate to convert it into acetyl CoA. The site of this reaction is matrix of mitochondria. But the point should be noted here that this is not a part of Krebs cycles, and the enzyme which helps in this conversion is pyruvate dehydrogenase complex, which is actually a group of 3 main enzymes and 5 coenzymes. The pyruvate will convert into acetyl CoA by decarboxylation and adding one coenzyme A, and NAD is reduced in a NADH. Now let's talk about the pyruvate dehydrogenase complex. That what are that 3 main enzymes and that 5 coenzymes. The first enzyme is pyruvate dehydrogenase E1, and the coenzyme which works along it is a thymine pyrophosphate. The second one is dihydrolipoyl transacetylase. The coenzymes are lipoic acid and coenzyme A. The third enzyme is dihydrolipoyl dehydrogenase, and the coenzymes are FAD and NAD. Now let's talk about the mechanisms of pyruvate dehydrogenase complex. The first step will be the pyruvate will go on decarboxylation process. In the presence of pyruvate dehydrogenase enzyme, that is E1, and by the help of TPP, this pyruvate will convert into the hydroxyethyl TPP, and one carbon dioxide is released from here. And again, on the presence of E2 enzyme, the lipoic acid is gonna attached with this hydroxyethyl TPP and make acetyl lipoamide, and the TPP is released. And again, the other coenzyme, the coenzyme A, is going to add on this acetyl lipoamide and form acetyl CoA and reduce the lipoic acid from that acetyl lipoamide molecules. But here the E2 is still attached with acetyl CoA. That's why the other enzyme E3, which is dihydrolipoyl dehydrogenase, is gonna perform the oxidoreductase reaction, and it will detach the E2 from acetyl CoA. By the help of FAD, it will oxidize the lipoic acid and make the E2 its in initial state and form FADH2, and another NAD again reduce itself and NADH, and at last we will found NADH. This pyruvate dehydrogenase complex is also known as a vitamin B complex, because all that coenzymes which are very important and plays crucial roles in this complex are derived from the vitamins B1, B2, B3 and B5. Let's talk about the regulation. It is directly regulated by the energy forms molecules like ADP, NAD, FAD, which is a low state of energy molecules. Low energy molecules is gonna stimulate the pyruvate dehydrogenase complex, and the molecules like ATP, FADH2 and NADH is going to inhibit the pyruvate dehydrogenase complex. The same way, the two proteins like protein kinase and the phosphoprotein phosphatase, the two enzyme is going to regulate indirectly. And the presence of acetyl CoA, ATP, NADH, this protokinase is activated and phosphorylate the E1 enzyme of this complex and inhibit this pyruvate dehydrogenase complex. On the other hand, in the presence of calcium, this phosphoprotein phosphatase is going to be activated and dephosphorylate the E1 enzyme, and again pyruvate dehydrogenase complex is stimulated. Now let's talk about some clinical conditions. The main two clinical condition is going to be occur if this enzyme is gonna be mutated or gonna be deficient. The number one is lactic acidosis and another is neurological deficits. Now there is one important disease, that's congenital lactic acidosis, which is X-linked genetic defect in which the pyruvate dehydrogenase complex is mutated or inherited and pyruvate is converted into lactate instead of acetyl CoA. And according to the severity, we can divide it into three types, like very serious, moderate case and milder case. In very serious case, we can see the neonatal death. In moderate case, the severe mental retardations Even death can occur in infancy. In milder case, when the patient will take a lot of carbohydrate, the patient will suffer from some neurological deficit like episodic ataxia. Then what will be the treatment in this condition? The treatment will be simply we have to reduce the carbohydrate from the diet of the patient, like ketogenic diet. Thank you.